Hello, hello. Thank you for keeping your attention. I know I'm the last talk, so I'll try to get through it quickly. Awesome. Well, hello everyone. My name is Maggie Hill. I'm from the University of Rhode Island, Rowley Laboratory, and today I'll be talking about Pseudoalteromonas rubra, a marine bacterium that packages Prodiginine antibiotics in outer membrane vesicles. So first I'd like to introduce coral as a holobiont, which is a term coined by Dr. Lynn Margulis in the 1990s, essentially describing eukaryotic organisms that have evolved together with microorganisms, commensal bacteria and symbiotic. And essentially, they've retained an ecological role but maintained their own genomes. And then also we have global warming, rising ocean temperatures, unfortunately, that cause these algosymbionts to be expelled from the corals, which can be depicted in this picture of a Stranger populata, seen in these bleaching white coral versus the healthy brown coral. So a Stranger populata is our coral of interest because not only is it the state coral of Rhode Island as of recently, but it's also very easily cultivated under laboratory conditions. And another key player in this story would be Prostose coralline algae, which is a red algae that is a known coral larval substrate for tropical corals, and we hypothesize that it can also induce settlement with a Stranger populata, which is a temperate coral from Rhode Island. So how does CCA affect the settlement of coral? And this one study looked at the microbiome and metabolome to compare different chemical cues, and they looked at 6 different species of CCA and how it affected the larval settlement of this particular coral. And they found that the one that had the most significant rate of larval settlement also had the most signature microbiome and metabolomic cues. So chemicals play a significant role when we talk about larval substrate settlement. And some previously identified bacteria-derived coral settlement compounds. I'm sure some people in the audience are very familiar with tetrabromoferol, which was originally isolated from a Pseudoalteromonas species, and essentially induces settlement and metamorphosis with many tropical corals. So now I'm going to talk about Pseudoalteromonas rubra, which is a prolific producer of prodiginine chemical cues. So these prodiginines are red pigmented, causing these cultures to be pinkalicious, one would say, hence the pink soup. And they've been tied by the Peter Shook group to induce larval settlement as well. So extracts produced isolated cycloprodigiosin, prodigiosin, and 2-methyl-3-hexylprodigine. And cycloprodigiosin has a significant impact on larval settlement when compared to the control of CCA. Another paper that they published also looked at the mono and multi-species biofilms to induce larval settlement, and I'd like to direct your attention to 2 strains, 1783, which would be Pseudoalteromonas rubra, and 1772, which is not Pseudoalteromonas rubra, but due to the genomic sequencing, has 99% similarity with Pseudoalteromonas rubra. So this Pseudoalteromonas rubra is very, very, can be tied to coral larval settlement. And I'd like to also bring another factor in. This is getting very complicated, but bear with me. And I'd like to talk about membrane vesicles. And if you're not familiar with membrane vesicles, essentially they're these protrusions from the bacterial cells that could either include the inner membrane, the outer membrane, and bits and pieces of the enzymes that could be present on either membrane. They also package DNA, RNA, and special secondary metabolites. And another group correlated these membrane vesicles with the settlement of another marine invertebrate, a Serpulidae. And this marine worm, when introduced to the wild-type biofilm of C. litica, they normalized that to 100% metamorphosis. When they used just the OMBs, that also induced settlement. And OMB extract, which is direct correlation to the secondary metabolites packaged within the MVs, that still induced settlement to some extent. So very important research, and keep that in the back of your mind going forward. So what are we interested in at the Rowley Lab? So one, we want to see if membrane vesicles isolated from CCA-associated bacteria, whether or not they mediate coral and microbial interactions. And if so, what are they packaging? So here's our hypothesis. We're assuming that microbes within this microbial biofilm atop the CCA here are producing bacterial membrane vesicles to recruit competent larvae, hopefully Escherichia populi, and that they contain specialized metabolites, potential chemical cues to induce this recruitment. So how do we test this? First, we isolated 2 CCA-associated strains of Pseudoalteromonas rubra, KB1 and CH007, and we created a workflow that could, 1, produce bacterial membrane vesicles, and 2, characterize them. So first, we're going to grow up these cultures and centrifuge down the cell pellet to remove that. So, and keep in mind, we're doing 3 fractions here. We're taking the cell pellet for subsequent extraction, and then once we have that filtered supernatant, we're going to subject that to ultracentrifugation, including a wash cycle, and that results in another fraction, which is the MVs, which are either resuspended in 1 times PBS with 1% DMSO for bioactivity testing, or pure millicue sterile-filtered water for chemical analysis. And we also are saving that MV-free supernatant for extraction as well. We're subjecting these MVs to characterization methods such as nanoparticle tracking analysis to determine our concentration as well as that average size and diameter. And we're using transmission electron microscopy to actually see our membrane vesicles in a micrograph. We're testing them for bioactivity and we're chemically analyzing them by tandem mass spec. So first we wanted to optimize this a little bit and hone in down on those prodiginines that I mentioned before. So we created a growth curve for each strain. The doubling times are very, very similar, as you'd expect. And we looked at the prodiginine content at each time point. And we found that it was significantly detectable. Although the strains do turn pink around 8 to 10 hours, it's not detectable in a milligrams per mil content until about 12 hours and then 24 hours. So we'll be comparing 12 and 24 hour MVs throughout this entire experiment. Here are some pictures. So we first have our micrograph here. This is KB1 12-hour MVs. This is representative of all of our batches. And if you notice that we see 1 dark circle around, those would be indicative of an outer membrane vesicle. And like I said before, these membrane vesicles could be outer inner or just simply outer. And if we saw maybe 2 rings, maybe they'd be outer inner, but it would appear that they're just outer membrane. And then we get our nanoparticle tracking analysis data, which would lend us a concentration that if we multiply by the amount of mils that we harvested, we get our total particles harvested, and as well as their mean size in nanometers. And I want you to look at the differences between the 24-hour and the 12-hour. So the